Dear students, welcome to our lecture number 37. Myself Dr. Anjana Rani, working as an assistant professor in Noida Institute of Engineering and Technology, and I am here to teach you protein binding. See, in the previous lectures I have told you about the complexation. What is complexation? What are complex compounds? What are its different applications? Okay, and what are the classification of complexes. Protein binding is also a type of complexation. Okay, so what are complex compounds? Complex compounds are the compounds in which complex compounds are defined as the molecules in which most of the bonding structures can be described by the classical theories of valency. Okay. So, these complex compounds are formed due to different type of interactions, and these interaction can be van der Waals force of attraction, this can be ion dipole interaction, dipole interaction, it can be hydrogen bonding, or there can be covalent bonding or coordinate bond formation between the molecules, so as to the formation of the complex compound, and the process of this complex formation is known as the complexation. Okay. So, these complex compounds possess different properties from the properties of the components. For example, we have two components A plus B, and these will make a compound AB. So, the properties of this AB compound will be different from the properties of these components. Okay. There can be enhanced solubility after the complex formation, or there can be enhanced bioavailability or reduced bioavailability. Okay. There can be a change in the physical state. Okay. We can convert any liquid compound into its solid state with the help of complexation. We can reduce the volatility of any substance with the help of complexation, or chemical stability can be enhanced or can be reduced by the complexation. This complexation has application in solubility, in dissolution, in the partition coefficient, in the absorption and bioavailability, in the toxicity, and also with the help of complexation process, any compound can act as antidote in case of metal poisoning. So, this complexation behavior has wide application in handling of dosage form or in pharmacy. So, now comes to the protein binding. So, the protein binding is what? The process of complex formation between the protein and the drug is known as the protein drug binding or protein binding. So, protein binding is also a type of complexation, and it has wide application in pharmacy. Okay. The phenomenon of complex formation of drug with protein is called as protein drug binding, and the proteins are particularly responsible for such an interaction. So, a protein drug binding form. Okay. A drug can interact with several tissue components. See, there are different types of protein which can bind to our drug. Most of the time there are two types of protein. One is albumin and second one is globulin. Second protein is globulin. Basically, there are two types of protein that can bind to our drug. Albumin, the level of albumin in the blood is very high, so it can bind to wide variety of drugs, but the level of globulin is less as compared to the albumin. Another protein is, another macromolecule is alpha acid glycoprotein. Alpha AGP. What is known as alpha acid glycoprotein. Okay. So this the level of this alpha acid glycoprotein in normal healthy individual is very less, but in case of any diseased state, for example myocardial infarction, the level of this protein is increased. Okay. And the protein globulin binds to the basic drugs. The albumin, this albumin binds to the acidic drug as well as the basic drugs. Okay. Two types of drugs, acidic and basic. Albumin protein binds to acidic as well as basic drugs. But AGP or alpha acid glycoprotein binds to basic drugs mostly rather than the acidic drugs. Okay. These can bind with acidic as well as basic drugs. Okay. But alpha acid glycoprotein binds more with basic drugs rather than the acidic drugs. Now comes to globulin. What is globulin? Globulin binds to the drugs up to a little extent. Basically the globulin interactions we can see in case of immunoglobulins. Globulin interaction are seen in immunoglobulins. Okay. Now binding of drugs falls into two classes. First is blood and second one is extravascular tissue proteins, fats and bones, etc. Okay. In the blood there are two things, one is plasma protein and second one is blood cells. Okay. Drug can bind to blood cells as well as to the plasma proteins. Okay. The proteins present in the plasma, and the protein that I have told you recently, that there can be albumin, globulin or alpha acid glycoproteins. Okay. The concentration of albumin and globulin is very high in the blood, so there are more chances of binding the drug to the albumin and globulin. But globulin interactions are seen in immunoglobulin. Albumin can bind to acidic drugs as well as to the basic drugs. Okay. And alpha acid glycoproteins binds only to the basic drugs, as means more extent to the basic drugs as compared to the acidic drug. Okay. So protein drug binding falls into the two classes. One is the blood. In case of blood, it can bind to plasma proteins and it can also bind to the blood cells. Next is the extravascular tissues, proteins, fats and bones. Okay. So this protein binding can be divided into two types, intracellular binding and extracellular binding. Okay. There can be two types of binding. One is intracellular binding, second one is extracellular binding. As the name suggests, intracellular means between the cells. Okay. Now comes to mechanism of protein drug binding. Okay. I am telling you albumin can bind to acidic drugs as well as to the basic drugs. Alpha acid glycoprotein only binds to the basic drugs. But how it binds to the drug? What is the mechanism behind this protein drug binding? What is the process of this complex formation, or what is the process of complex formation between the protein molecule and the drug molecule? Okay. So binding of drug to the protein is generally of two types, reversible as well as the irreversible. Okay. the drug protein binding can be of two types, reversible and irreversible. Okay. So, reversible type of binding involves the weak chemical bonds. The reversible generally involves weak chemical bonds such as hydrogen bonds, hydrophobic bonds, ionic bonds and van der Waals forces. Okay. There can be these different types of interactions between the reversible types that will help in the protein drug binding. Okay. So, next is the irreversible binding. What is irreversible? Irreversible type of protein drug binding is very rare and it can result by the covalent bond formation. Okay. It can arises as a result of covalent binding and is often a reason for the carcinogenicity or tissue toxicity of the drug. Okay. So, what I have told you, there are two types of protein drug binding mechanism. There are two types of mechanism which are associated with the protein drug binding. One is the reversible, second one is the irreversible. Okay. Reversible mechanism includes hydrogen bond, it includes hydrogen bond, hydrophobic bond, van der Waals interaction, and also ionic bonds. Okay. Next is irreversible type, and this irreversible type consists of covalent bond. Okay. So, there are two types of protein drug binding mechanism. One is reversible and second one is irreversible. In the reversible mechanism, there is hydrogen bond, hydrophobic bond, van der Waals forces of interaction and ionic bonds are involved. But in case of irreversible mechanism, covalent bond formation occurs and this covalent bond formation or this covalent binding can resulted in the carcinogenicity or can be a reason for the carcinogenicity or tissue toxicity of the drug. Okay. Now, the factors affecting protein drug binding. For the factors affecting protein drug binding includes, first is drug related factors. Okay. The drug related factors will include the properties of drug, the amount of drug, the quantity of drug or the affinity of the drug. Okay. So, drug related factors include physico-chemical characteristics of the drug, second is concentration of the drug in the body, third one is the affinity of a drug for a particular binding component. Okay. So, firstly physico-chemical characteristics of the drug. Physico-chemical means physical as well as the chemical properties of the drug. Means protein drug binding will depend upon the physical and chemical properties of the drug. Second is concentration of drug in the body. Okay. Protein drug binding will also depend upon the concentration of drug that is present in the body. Third is affinity of a drug for a particular binding component. Okay. As I have told you that certain macromolecules or certain proteins have interaction to the drugs. For example, I have told you albumin can bind to acidic drugs as well as to the basic drugs. but alpha acid glycoprotein mostly bound to the basic drugs. So, this is all about the affinity. Okay, albumin has affinity, or acidic drugs and basic drugs have affinity for binding to albumin protein, but the basic drugs have more affinity for the alpha acid glycoproteins. Okay, these binding are due to the affinity of the drug for a particular binding component. Okay. Next is the protein or tissue related factors. Okay, the protein or tissue related factors include two factors. One is physicochemical characteristics of protein or binding agent. Second is concentration of protein or binding component. Okay, so physicochemical characteristics means physical and chemical characteristics. Okay, so the protein drug binding will also depend upon the physical and chemical properties of the proteins or the binding agent. Second is concentration. The amount or the concentration of the protein present also affects the extent of protein drug binding. Next is the drug interactions. Drug interaction include three factors. One is competition between the drugs for the binding site. Okay, see, there are specific numbers of binding site, and suppose 2 or 3 types of drugs are available for binding to the sites. So, there can be a competition between the drugs to bind that particular site, and this resulted in the displacement interaction. Okay, the drug which have more affinity towards the binding site will bind to the drug. Okay, and if another drug that we have administered has more affinity as compared to the previous drug, that in that case this drug will displace the previous one drug and bind to the binding site. So, this is the displacement interaction. Okay, suppose this is the protein and a drug has affinity to bind to this. So, this will result in the formation of this complex. After some time, if a drug B is administered and the affinity of B to bind at this particular protein is more as compared to the drug A. If affinity of B is more than A, in that case this B drug will displace the A from this and resulted in the formation of complex BP. Okay, understood? So, this interaction will be known as the displacement interaction. What happens in this case? The B drug has more affinity to bind to this protein as compared to A. So, it displaces the A from this complex and bind to this P and resulted in the formation of this type of complex. So, this is known as the displacement interaction. Next is the competition between drug and normal body constituents. Okay. Third is the allosteric changes in the protein molecule. Sometimes due to this protein binding, there can be some changes in the structure of the protein molecule. So, if there are any changes in the structure of protein molecule, that can also affect the extent of protein drug binding. Okay. Next is patient related factors. Some factors are related to the patient, that can depend on patient age. Inter-subject variation means different subjects has different properties. Okay. So, inter-subject variation means the extent of protein drug binding will differ from subject to subject or person to person. Third is the disease state. In case, for example, the level of some proteins in case of disease state will vary. So, in that case, the level of protein drug binding will also affect. In the starting of lecture, I have told you that level of alpha acid glycoprotein, alpha AGP level is very low in case of normal healthy individual, but in case of disease state, the level of this protein will be very high. Okay. So, if the person is in disease state, in that case the level of this protein will be very high in the blood. So, there are more chances to the protein drug binding to this. There are more chances that drug can bind to this protein. Okay. So, in this way, in the case of disease state, the level of protein drug binding affects. So, these are the various factors that affect the protein drug binding. What are these factors? First one is drug related factors, second is protein or tissue related factors, third one is drug interactions, and fourth one is patient related factors. Now, what is the significance of this protein drug binding? What are the various applications of this protein drug binding? Okay. First one is the absorption. Okay. See, if a drug molecule binds to the protein molecule, then this is the protein molecule and this binds with this drug molecule binds with this protein molecule, then there will be a formation of complex, and if the size of this complex is high that it can't cross the GI membrane, in that case there will be a reduction in the absorption. Okay. So, the protein drug binding can affect the absorption process. Okay. I have told you in the very first class that these complexes affect the absorption process, distribution, elimination. Okay. So, protein drug binding is also a type of complexation process. So, this protein drug binding will also influence the absorption process. Okay. Second one is systemic solubility of drugs. Due to the complex formation, the systemic solubility of drugs will be reduced. Okay. Third one is the distribution. Okay. Drug protein binding will severely affect the distribution of drug. Okay. Protein drug binding decreases the drug distribution in the body. Why? Because protein bound drug is big in size. Okay. The size of the molecule will increase after binding to the protein, and if the size is high, then it cannot cross the membrane. It cannot cross the cell membrane. So, its distribution is restricted. Okay. Only free drug or unbound drug can only pass through the membrane and contributes to the tissue binding. Okay. So, higher the protein binding, lower it will contributes to the tissue binding. Okay. Higher the protein binding, lower the tissue distribution. Okay. And this tissue distribution we can express in the form of volume of distribution. Volume of distribution means the volume in which our drug molecule is distributed. This is known as the volume of distribution. Okay. If the size of protein bound drug is high, then it cannot cross the cell membrane and its distribution will be restricted, and due to this restricted distribution, the volume of distribution very low or the tissue distribution will be very low. Okay. Next is the tissue binding, apparent volume of distribution and drug storage. So, the drug protein binding has also application in case of tissue binding, apparent volume of distribution and drug storage. As I have told you that if the protein binding is high, its volume of distribution will be less. Next is the elimination. Protein binding will decreases the renal excretion of the drug and enhances its biological life. Next is displacement interaction and toxicity. Okay. Displacement interaction means if a certain drug has more affinity as compared to the previous one drug, then there can be displacement interaction. The drug can displace the previous one drug and binds to the protein drug molecule. Next is the diagnosis. See, this protein drug binding is also useful in case of diagnosis of various diseases. Next is the therapy and drug targeting. So, this protein drug binding has also significance in case of treatment and in case of drug targeting. Certain drug molecule or certain substances we can bind with the protein that helps the drug to reach to the receptor site or to the action site, so that it can deliver its action. Okay. So, this protein drug binding can also act as a carrier system. Okay. So, protein drug binding or protein drug complexes can act as the transport system to carry the drug to the site of action. Okay. Another significance is sustained release. See, the complex of drug protein in the blood can act as a reservoir and continuously supply the drug to the action. Okay. So, this is all about the significance of protein drug binding. So, in this lecture what we have discussed, what is protein drug binding, what are the types of protein drug binding, what are the various mechanisms associated with protein drug binding, and lastly what is the significance of this protein drug binding. Okay. In the next lecture we will discuss the kinetics of drug protein binding. Okay. Thank you.